Okay, hi everyone. Today we're going to be looking at chapter 5. So this chapter 5 is the sample preparation for inorganic analytes. Okay, so basically the sample preparation is going to be involving only spectroscopic methods, which the one that we have learned the previous three chapters, basically two chapters of atomic spectroscopy, atomic absorption spectroscopy, atomic emission spectroscopy. All right. So basically now to look at the how do we prepare the sample after we have done the sampling process. And then you still remember that during our first lecture, if I'm not mistaken, that we have looked into how to sample, how to do the sampling process, how do we preserve it, and how do we store our sample prior to analysis. So now we are going to prepare the samples for the analysis of the specific spectroscopic methods. Okay, all right. So basically, first of all, you need to know on the type of equipment in terms of atomic spectroscopy that you are going to be using. All right. So if you are using flame AAS or if you are using graphite furnace AAS, maybe the sample preparation is going to be minimized because you just put your sample inside the graphite tube. Okay, if you still remember the diagram of a graphite furnace and the advantages of a graphite furnace, it is the sample preparation is minimized. But for flame AAS, the sample preparation you need your sample to be in a solution form, right? Okay, so you need to know whether you want to do flame AAS, graphite furnace AAS, even ICP OES and ICP-MS. So you need to know on that first. All right. So, yeah, we are going to be back to these figures. As I mentioned, this is a very important figure in terms of how are you going to choose the instrument based on your ends. Okay, for example, if we looked at the highest end here, when you looked at ICP-MS, it is the most expensive but has the best detection, which is until part per billion. Covers almost including hydrides and Hg. It is a fast multi-element analysis. So basically, for this ICP-MS, yes, it is the best. It has the lowest, that it has the best detection limits. It can detect almost all of the element, but it is very expensive. Okay, but if you go back to the extreme, which is the flame AS, it is cheap. Yes, it's cheap. The detection limit is only up until ppm level. Fast per sample, but a single element analysis. So as we mentioned before, a single element analysis for flame AS to know why does that happens. The light sources of HCl, hollow cathode lamp. All right, but for ICP-MS using plasma, so all of the elements have been detected simultaneously. So back to this decision criteria, you need to know, you need to choose on which on which instruments that you are going to be using. Okay, so yes, so for this one also, it is the same criteria in terms of number of analytes and the detection limits. Okay, all right. So yes, we are back to this table of when we are looking at into the methods of flame AS, graphite furnace AAS, even hydride generation AAS, ICP OES, and ICP-MS. So basically, all of this are important in terms of what instrument are you going to choose. All right. So, an example of heavy metal analysis. So now we are going to look at into the critical points of heavy metal analysis. All right, so because heavy metal analysis, before this, we have looked into the applications of flame AAS, even graphite furnace AAS, hydride generation AAS, even in ICP, OES, and ICP-MS, all of those instrumental methods are pertaining to the more or less towards heavy metal analysis. All right. So first of all, when we looked at into the instruments, the instruments is at this caliber. Okay, at the end of the analysis. Okay, kalau kita tengok in terms of the heavy metal analysis, the instrument is at the bottom part, which is it is the instrument to be used to analyze. So what happens before the instrument before the sample can be used for the analysis. All right. So basically, when we looked at into the sampling and sample pretreatment, all right. So the critical points here that you need to understand is that when you do sampling or sample pretreatment, you need to have an idea on how to avoid contamination when preparing a test portion. All right. In terms of avoid contamination, you we have discussed this before during our I'm not sure, but I think it's about the second chapter, if I'm not mistaken, that in terms of avoiding contamination, that for your sampling, maybe for your if you are doing a sample TIFF for a liquid sample, when you do the sampling process, you can always rinse it with 5% or 3% of um, nitric acid for to avoid contamination, to avoid cross contamination from the previous sampling process. So you can soak it into the nitric acid for overnight prior to your sampling process the next day. All right, and then after you have done that, when you go to the sampling site, you can always rinse it with the matrix or with the if you are collecting or you do the sampling process at the for example river A, so you can wash your sample teeth using river A to make sure that it is free of the previous previous sampling process. All right, so first of avoid when preparing a test portion. Contamination here means that contamination may be from the previous sampling process. You don't want to have a cross contamination. The second one is to avoid is to obtain homogenized representative sample. So basically for this one, you need to able to select on which depth are you going to select for your test sample. Homogenized representative test sample. All right. So usually for heavy metal analysis, it is just below the surface. Or if you decide to go maybe 5 or 10 meters below the surface, then you need to do just that. It depends on your method. Okay. All right, so that is for sampling and sample pretreatment. In terms of sample preparation or digestion, so this is where you are going to learn now. Okay, so you need to use digestion according to the type and amount of sample. So we have different types of sample. If you have a liquid sample, then you have a different type of sample preparation or digestion. If you have solid sample, that's going to be different, right? So we are going to look this in depth after this, all right, for this one. And then the second point is to avoid contamination of. This is why uh, so your sample tips soak before, I mean 1 or 2 days before your sampling process, you need to rinse it as well with the river water on your sampling site. All right. So and then this is for to avoid contamination lah in terms of maybe excess acids or excess dust inside your sampling teeth. All right. Carry out blank tests in parallel by the same procedure as well. So basically, you need to have um, a blank test as well lah. Okay, to make sure that your process is okay. All right. So after your sampling, after you have done your sampling, you've done your pre-treatment as well as you have done your sample preparation, then you need to prepare preparation of test solution. All right. So this is after you have done your sample preparation in terms of digestion. Okay, you need to check whether your digestion is complete or not, whether you have achieved 100% digestion. All right, and then you need to prepare the test solution with a similar concentration as those of the calibrants depending on the ICP-MS or depending on the spectroscopic methods that you have done. So basically you need to prepare your sample according to the spectroscopic method you have chosen. All right. All right. Lastly, you need to know in terms of the determination of the calibration. Alright, so this is something that is for the instrumental punya part lah. So make sure that your instrument has been calibrated yearly so that your results will be much more reliable. So that's why we said here that instrument needs instrument checkup lah basically. So you need to look at your performance of the instrument itself. Alright, so these are the critical points that you need to know in terms of when you do or when you think about your analysis. All right. Okay. So moving on. All right. So sample preparation basics. All right. So these are some of the definitions. What do we mean by sample preparation for AAS or ICP? Basically, it's just the conversion of a solid or a liquid into a liquid that is suitable for AAS or ICP measurements with a pneumatic nebulization. All right. So for AAS, for ICP, you already knew that it needs to be in a liquid form because it has a nebulization process, which is converting the liquids into aerosols. So basically, based on that, you straight away think that, okay, I need the sample needs to be in a liquid form. It doesn't matter if it is a solid prior to the analysis. You need to get your solid into a liquid. All right, but we will look into it after this. Okay. All right, into what is the sample solution? It is a liquid form of the sample. So the preferences is having the analytes in an ionic solvated form or observing analytes with a 0.3 microns filter. All right, so basically this something that what I have mentioned before, you convert your sample into a sample solution. It doesn't matter if your sample is solid, but you need to convert it into a sample solution. Right? What is a standard solution pula? Okay, standard solution is a liquid form of a known concentration of the calibrated and quality control standard. Okay, now when we talk about standard solution, all right, for example, you want to measure or you want to quantify later on for copper. Okay, so copper standard needs to be in a solution form. Standard here means that you know the concentration already. So you say that, okay, I have a standard of 100 milligram copper in a solution lah. So basically 100 milligram of copper in 1 liter of solvent. So you know the concentration, while for your sample solution, you know that there is a copper inside, but you don't know how much is it. You don't know the concentration. All right. But for standard solution, when we are talking about standards, you know you know what is the identification of it. All right. All right, and then the last one is matrix matching, is the ability to match the sample solution to the standard solution such that all nebulizer and plasma related effects are eliminated or can be corrected. All right. Okay, now we are going to look at into the sample preparation. All right, sample including sample pretreatment is a step in chemical analysis where the sample is brought into the correct size or form for analysis. Okay, talking about sample preparation is it is not just you preparing your sample for the analysis. Okay. Now would you imagine that, okay, for example, you imagine that you want to analyze heavy metals in a fish. Okay. So basically, you don't want the fish, all part of the fish to be converted into solution. When converted into solution, kita analyze through our, for example, AAS. All right. So you need to determine the correct form of the sample itself to fit for the analysis. All right. So the general principles is that you need to first of all determine the amount. You need to know how much or where, basically. For example, okay, imagine now we have a dead fish that we want to analyze the heavy metals. So where is the best place to get the sample? Okay, now you decide based on the standard method. When you have read the standard method, maybe it says that okay, 10 gram of the fish. So where do you want to get this 10 gram? Is it by the tail? Is it by the head? Or is it by the body of the fish itself? Right? So you need to determine the amount, the weight or the volume. And then you also need to have replicates samples. So when you read that, okay, I need to have 10 gram. For example, when you say that 10 gram from the body of the fish. So you don't just prepare 10 gram, you need to prepare at least 30 gram, which is 10, 10, 10. You need to prepare some sort like a replicate, which is in this case, usually in chemical analysis, we have triplicates. You have to have triplicates amount or triplicates of the same amount of your sample. All right. So these replicates will be analyzed the same way, will be subjected to the same pretreatment, will be subjected to the same analysis at the same time, so that the deviations would be the same, and then at the end we can have the mean lah. Okay, we can have the average of the results. So, all right, that's the second general principles of sample preparation, right? And then the third one, we need to bring the analyte. So in this case, kalau contohnya heavy metals, you need to, for example, you want to detect arsenic. Okay. So bring the analyte, okay, bring the arsenic into the best chemical form for the assay method used. Best chemical form here means that, okay, for example, kalau if you use graphite furnace, what is chemical form? It can be anything. It can be solids, it can be liquids, it can be slurry. But if you are using flame AAS, what best chemical form? It needs to be in a, so it depends on your it depends on your sample, so it depends on your instrument of that you desire to use. Then based on the instrument, what is the best chemical form, right? And then the fourth one is we need to bring the analyte, which is in this case, for example, arsenic, into the best concentration range for the chosen methods. This is also depends on your instrument that you choose. If you have your analyte at the ppb level, part per billion, for example lah, we have at the part per billion level. So you cannot use flame AAS because flame up until part per million, you need to use ICP-MS. So this apa orang kata, this thing in terms of best concentration range, you need to be able to think about. All right. So if you are able, for example, the arsenic level is quite trace, quite low, always pre-concentrated it. Okay, when you have at ppb level, part per billion, you can always increase the concentration. There is a way of doing that for it to become part per million. We call it pre-concentration. So how to do that? And when you have part per million, you can use flame AAS. So that's why I said the best concentration range, right? And then the last thing is that we need to pre-treat, separate, pre-concentrate, and eliminate interferences. So are the objectives basically of sample preparation. We need to pre-treat, separate. For example, tadi kan in terms of the solid part of the fish, we need to pre-treat the solid part of the fish. We need to separate them. So you don't want all the solids. You only want the solutions. You only want, because when you do your pre-treatments, your can imagine that your heavy metals solution. So these solutions you need to separate from the solids, which is the isi-isi kan tu, and then you need to pre-concentrate, eliminate the interferences. All right. Okay. So the good laboratory process for sample preparation. So the good process for sample preparation basically the management of analytical blanks in terms of the cleanliness of the laboratory environment, because you need to know that you need to avoid cross contamination. You need to avoid that because this is for this GLP good laboratory process. Good laboratory practices, good laboratory practices, it's important for you to have this GLP during the preparation of trace elements, mainly specifically trace elements, because trace elements you tend to, I mean that if you didn't have a GLP, then your trace elements sometimes it cannot be detected, or even some interferences can go into your sample. All right. So analytical blank is the measure of all the external sources of elemental contamination, including the digestic acids, and make corrections to the measured sample reading. All right. When you have your digestion beforehand, you use acids, you use all sorts of other matrices to digest, to prepare your samples prior to the analysis. So all of this would have some sort like a deviation, uh, even may increase or decrease the concentration of your trace element analysis lah. Okay, so contamination for trace analysis can occur from the laboratory environment. So your for a GLP, so you need to make sure that we have clean air facilities, we need to have air filters, and you need to have you need to do your work inside the fume hoods. Okay. All right, in terms of materials, we have plasticware for storage and handling. Some plastics might seep back into your analyte, seep back into your solution. So we'll introduce a new interferences, right? Reagents, apparatus, and then lastly is the analyst. Analyst itself, the skills of being an analyst is also part of GLP. All right, so basically when you think it with your bare hands, you don't know what apa things that are available at your bare hands in terms of cosmetics, hair, wearing jewels or watches, which all of the analytes, all of the interferences would seep back into your sample. All right. All right. So, sample pretreatment for solids. So, for lecture, I will just go into the solid dulu lah. Okay. Let me look first. All right. So maybe I just go into the solids. All right. So sample pretreatment for solids. So we have green and sieve, which is we need to obtain a suitable size, which require in terms of the homogeneous size lah. So we don't want some solids have bigger sizes, some solid have much more smaller sizes. So we need to have size, a homogeneous size. Okay. And then in terms of homogenize, the solid sample is placed in a blender with a solvent. For example, well, you put your sample in the case of fish, you take the fish, the flesh of the fish, you put into the blender, you put your solvent in, and the sample is homogenized into a finely divided particles. All right, so this pretreatment is useful for plant or animal tissues in terms of food and the environmental samples lah. All right, so these finely divided particles promote more efficient extraction. Why? Because finely particles has much more surface area. So much more surface area, the extraction solvent would be able to extract much more higher yield as compared to a bigger blob of your sample. All right. So after we have done the green and sieve, and then we dry the samples to remove the moisture. All right. So all of the, for example, biological samples, we have some water content in it. So we need to remove the moisture as much as possible because this um, might apa might decrease the concentration of the extraction yield. All right. So dry the samples to remove the moisture about 105 until 110 Celsius for about 1 or 2 hours lah. All right. The side reaction or decomposition of samples must be considered also. All right. Following by storage in a desiccator. So desiccator ni is an enhancement. It's just that a further drying process. All right, you have dried your sample inside an oven and then you put into your desiccator to remove more moisture. All right. Okay. So these are the samples and the drying conditions that you can have a look. So the sample preparation after we have done drying it, so we need to solubilize the sample in solution. So basically this is a destructive where harsh methods that destroy the organic components that constitute the sample matrix. Alright, organic analyte and inorganic matrix remains after dissolution. Only organic for example like in our fish sample, the lipids of the oils inside fish would be apa orang kata destroyed, but only inorganic analyte and inorganic matrix will be remain after dissolution. So how are we going to achieve this dissolution? So we have four methods, which is simple dissolution, where the solid is dissolved in a solvent without chemical change. Second one is dry ashing, where we oxidize by slowly heating in oxygen at very high temperature, about 400 to 700 Celsius, yeah, in the furnace. The third one is wet digestion, which is an acid treatment. We heat the sample with mineral acids in open or closed container in a microwave digester. And the fourth one is a fusion technique where we heat with flux of or acid until it reaches molten state. Okay. All right. So for dry ashing, as I mentioned before, it uses the furnace at temperature about 400 to 700 Celsius. So this resulting ash, which is free from organic compounds, is dissolved in dilute acid. So basically, you have dissolved the organic compounds. Now it left with only just inorganic analyte and inorganic matrix. So these inorganic analytes and inorganic matrix will be dissolved in dilute acid, right? So ashing usually, as I mentioned, at this temperature, and then it decompose the organic matrix, resulting in inorganic residues, right? So these temperatures can be too high for volatile metals such as mercury, arsenic, selenium, and lead. So other metals such as tin may form insoluble refractory compounds during dry ashing. So as I mentioned before, for your sample, uh, 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 for your sample for hydrate generation can for your elements, there are some elements that can form hydrides, which is some elements that can be volatile that can form hydrides.